Once again, a very warm welcome to the final webinar of the PREPARE project. It's been indeed a while now that we started PREPARE, 7 years back, and so it's really a great honor to co-moderate this final PREPARE session together with Jeremy Farrar, Director of the Wellcome Trust. And so I will start highlighting the successes and also the challenges of prepare and recover, but I will particularly focus on the lessons learned with regard to pandemic preparedness. And then Nina will talk about the rapid deployment of research in public health emergencies and some of the challenges that she has encountered over the last years. Marion will talk about diagnostic preparedness. Mark will talk about the future of clinical research in Europe, and particularly what our vision is with regards to eCRATE. And then the final session will be moderated by Jeremy. So when I started thinking about my talk, I realized that I kept or I'd stored an article that was published back in April 2014, so 7 years back, and it was actually the first major interview of Jeremy Farrar as the director of the Wellcome Trust in The Guardian. And I don't know why I kept this article, but when I was rereading it over the weekend, I realized that he had spoken about novel coronaviruses as a potential threat next to drug-resistant pathogens. And I was amazed to see that already at that time, novel coronaviruses were on the table as a potential threat for emerging infectious diseases. But I was also particularly struck by the comment that Jeremy made about the fact that the systems that we have got in place now are not fit for purpose when indeed the situation is moving quickly, as we have seen over the last 14 months. And that we have nothing that enables us to respond in real time. So that was what Jeremy said at his first interview in The Guardian in April 2014. Another interesting paper or another interesting study or an interesting study was published by Peter Horby's group last year, looking at the publications of a time describing treatment for patients with influenza H1N1 pandemic between the 1st of April 2009 and the 10th of August 2010. And Peter's obviously thought about this because of the threat with COVID-19 and challenge of doing clinical trials. And so it was very interesting to read that paper. And basically what he said was that published after the end of the pandemic, after WHO decided to call it an end to the public health emergency of international concern. And so in fact, over 33,000 treatment courses were started. Only 592 patients were included in interventional trials. And this is a very important comment in the paper as well. It was 8 months before the first interventional trial began recruitment. Think about this 8 months, because I will come back to this when I talk about the response in the clinical research response in PREPARE. And the other important conclusion from the paper was that none of the results were available during the pandemic, once again, between the 1st of April 2009 and the 10th of August 2010. So this is how, these are the challenges that we have encountered in the past during pandemics. And that is that the clinical research response to infectious disease outbreaks are indeed usually fractured and fragmented, and they're often delayed. Contrary to public health responses and also preclinical research response. And this came back each time when we confronted with emerging infectious diseases and with pandemic threats. And so the whole idea in PREPARE was to fast forward clinical research during an epidemics to improve clinical management. And so not only to improve clinical management, but also to reduce the size of the waves. And so those were basically the objectives of the PREPARE project. So it was funded from February 2014, and funding ended actually a month back in February 2021. And so PREPARE is an acronym for Platform for European Preparedness Against Re-Emerging Epidemics. We have close to 30 partners from academia, clinical networks, industry, professional organizations. I'm the coordinator of the project together with Menno de Jong in Amsterdam. And so the mission from the very beginning was to establish this PREPARE project as the European Clinical Research Framework. And I will just read the mission to you because it basically says everything. For harmonized large-scale clinical research studies on infectious diseases, prepare to rapidly respond to any severe infectious disease outbreak, and then with two objectives. On the one hand, to provide real-term evidence for clinical treatment of patients, on the other hand, for informing public health responses, and basically to close that gap between clinical research and public health. So this is the picture of the, let's say, PREPARE family. When we kicked off the project on the 7th of February 2014, on a gray day, and so this is the whole PREPARE family at that time. Most of the family members are still on board, and I have to say, when I look at this picture, it gives me very warm feelings because it's a really it's a great family, great colleagues, great scientists, and many of them have also become great friends. So it's been really a privilege to lead this project. There is, however, one person missing on this slide, and that is the person who basically had the whole idea of setting up the PREPARE concept, who managed to convince the Commission to invest in pandemic preparedness. But then, unfortunately, when we kicked off the project in 2014, became the director of the Wellcome Trust. So, Jeremy, it was very unfortunate, of course, for us that you had to leave the PREPARE consortium. We've missed you a lot, but we're also extremely grateful to everything you've done for PREPARE. And we know that you always supported us throughout the whole PREPARE lifetime, if I may say. So thank you very much for that, Jeremy. So in PREPARE, we focused on several types of clinical studies, observational studies, as well as adaptive platform design studies. And these studies have been discussed over the last 4 weeks or so during the webinars on each time on a Wednesday. And so these are the observation studies of the MERMAID studies. And again, I will not discuss them in detail. You can find the webinars on the PREPARE website. And then we also decided to build platform trial studies, the ALICE study and the REMAP-CAP study. So both of them were designed to become adaptive platform trial studies with response adaptive randomization. For both of them, the machinery was developed for such platform studies, but for ALICE, we were not able to roll out this machinery for reasons that were beyond our control. However, REMAP-CAP was launched successfully in 2017, and when it also became a global initiative. And again, you'll find all the data on REMAP-CAP, ALICE, but also in the MERMAID trials on the PREPARE website. What we also did was to develop a concept of responding to outbreaks or outbreak threats, if I may say. And so we developed these, let's say, so-called outbreak response modes, and divided them in 3 steps. Step 1 is when there's a limited threat to Europe. Step 2 is when there's a potential threat, and then step 3 is when there is an immediate threat and we need to start clinical research. So this is the concept that we developed during the course of PREPARE. And during step 1, the idea is to identify the research gaps and start writing the proposals in accordance with the research gaps. But that's it. And then in step 2, you start basically preparing for the trial, getting approval, for instance, from ethics committees. And then in step 3, we basically push the button and start enrolling patients. So that was a concept that we developed in PREPARE. And so then when the COVID-19 pandemic started last year in January, we were, of course, confronted with this potential threat for Europe. And you can see that we rapidly responded by already launching or kicking off step 1 on the 22nd of January. And in fact, on Sunday, the 19th of January, the Standing PREPARE Committee with Marion Koopmans, Peter Horpy, Menno de Jong, and myself already decided that this is a potential threat for Europe and could indeed become a pandemic. And so we already launched Mode 1 on the 22nd of January. It became very clear to us that the threat was emerging and became even stronger. And so on the 6th of January, we pushed the button for Mode 2. And then, of course, we lost a month. And we lost a month because PREPARE had come to an end. And so we didn't have funding or enough funding for the observational studies and certainly not for the interventional studies. And so we had to write a grant to get additional funding and eventually were successful and then received 5 million euros from the Commission for the RECOVER project. And so basically RECOVER was PREPARE's outbreak response action to the COVID-19 pandemic. Now, with PREPARE, we not only did clinical research, but also preclinical research. And for instance, one of the landmark papers was published already back in January by Christian Drosten and many other colleagues, including us, the PREPARE consortium. So this was indeed the landmark paper describing the RT-PCR for detection of the 2019 novel coronavirus. So I think indeed one can say that preclinical research response was very rapid next to the public health response. For clinical research, I think we were also very successful. And again, I would invite you to listen to the webinars on the PREPARE site, having a trained and collaborative network in place that allowed us to respond very fast to the EU calls for funding in response to COVID-19, because indeed the EU moved very quickly publishing these calls, and because we had a network, we could rapidly respond to that call. Having also a set of pre-arranged protocols on CRFs, having one of the largest multi-country patient cohorts on acute respiratory infections and arbor-like illnesses in Europe, and then, of course, very importantly, having a perpetual observational and interventional studies on ARI capable of enrolling these COVID-19 patients right from the very start of the pandemic. So I think this is an interesting slide, looking at the cumulative global recruitment of REMAP-CAP, because as I said, REMAP-CAP started as a PREPARE initiative, but then became globally in 2017, 2018. And in fact, it's actually, when you look at it, it's quite amazing that the first patient was already enrolled in this interventional study 38 days after the public health emergency of international concern by WHO. And so this is actually remarkable. On the other hand, we have to be honest also that perhaps this was pure luck. Why am I saying that this was perhaps pure luck? Because at the time when the pandemic started, we had slowly started enrolling patients in REMAP-CAP, starting the first patient on the 8th of April 2018. And because we had a delay in starting REMAP-CAP, we asked for an extension from the Commission for another 2 years. And so when the COVID-19 hit the world, we were still recruiting patients in REMAP-CAP. And that has been the reason why I think we were very fast in adding the pandemic stratum, and also very fast in enrolling the first COVID-19 patients on the 9th of March. And actually, we started enrolling patients during the first wave, but we didn't have enough funding. And it was only until June 2020 that the Commission informed us that there would be a possibility for a top-up funding of 50 million euros for REMAP-CAP. And then, of course, everything went even faster. And actually, the first paper was published already in September in JAMA on steroids. So I think it shows it gives one important message, and that is that you need to have infrastructure in place. You need to be able to, or need to have this perpetual trials to rapidly start when you have a pandemic situation. So let me move on quickly then with the 10 lessons learned. The first one is really about investment in pandemic preparedness and response. And during those 7 years, I had a lot of discussions with several people asking me, why do you get 24 million euros? This is a lot of money for pandemic preparedness. Well, I think the best answer to this now is to paraphrase what Mary Lasker was saying, that if you think preparedness is expensive, try the disease. And let's see after this pandemic, what was being the mental and the economic cost of the COVID-19 pandemic. And I don't think we'll have more discussions about that pandemic preparedness is expensive. The second lesson is really about the research response. And here I would like to paraphrase what Bill Clinton was saying. It's the science, stupid. And I think basically what I want to say here is that to me, the COVID-19 pandemic, it's really a victory of science, and it's really a victory of the scientists. And it's just absolutely remarkable how much research has been done, and wonderful data have been presented by the researchers over the last 14 months. And I think this is really, that's what I'm saying, it's really a victory of science, and it's really the science that has made a very big difference. And I honestly had not expected such a huge mobilization of so many researchers during the last 14 months. And I think what is also quite amazing is that these medical researchers were not only very productive and very successful, but they were also active in mainstream media and also on social media. And I think it was great to see so many researchers to present the data, but also to confront, I should say, xenophobia, and also to confront a populist hostility. And I think researchers should be really proud not only of what they've done over the last 14 months, but also that they were willing to confront what we often saw on social media, for instance, this populist hostility. The third lesson is about clinical research response. I think the research community, as I said, really responded admirably at an unprecedented rate to set up clinical trials. But I think it's also fair to say that we missed the bulk of the first wave. And I think that clinical research response was certainly fast, but it could have been faster. And I think it wasn't fast enough. But I think there was also one country where there was an exception, and that is the UK. And I'm really happy to discuss why the UK was an exception and what lessons we could learn from this, particularly for the European Union. But let me first give you the facts. So according to data from the COVID-19 Trials Tracker project, the number of clinical studies in COVID-19 up to August 31st, 2020 was close to 5,000. Close to 3,000 were classified as interventional trials, and 345 of them were randomized controlled trials. Of these 345 randomized controlled trials, 175 were related to drugs, of which 89 at phase 3 and 5 trials at phase 4. If you look at the number of entries, a lot of them came from France, nearly 500, and from Germany, about 245. And only 25 trials planned an enrollment of more than 1,000 patients, and 4,040 studies did not report at all a pre-planned sample size. And I'll come back to this. So what about the UK approach then? Because they were indeed very successful during the first wave. And what are the lessons learned for the EU? Well, in the UK, there was a national infrastructure for healthcare and clinical research in place with proper resources. There was a single UK-wide process to prioritize COVID-19 research, and only studies with an urgent public health designation were eligible for the NIH support. They had a standard contract, and there was no room for local negotiation. There was a simple protocol. Very importantly, there was top-down support from the chief medical officer, and again, I'll come back to this. And then finally, research funding already provided to UK hospitals was to be used for studies selected top-down. So I think there are some very important lessons here for the EU. Now, the EU is not one country. Nevertheless, and I'll come back to this, important lessons here for us for the future. The fourth lesson is about global clinical research response. I think it's fair to say that there was a lack of global leadership and lack of coordination of the clinical research response, certainly during the first wave. But on the other hand, we've learned that lesson, and there was a much better coordination during the second wave. However, research priorities, processes of publications to calls and selection of proposals, and procedural requirements were not aligned amongst the public funders. Another important lesson, and we need to do better in the future. Then about EU clinical research response. That was too fragmented, too fractured. For instance, there was no top-down prioritization of clinical research questions. There was a lot of competition between therapeutic agents, particularly about hydroxychloroquine and lopinavir/ritonavir. And this had, of course, an impact on enrollment of patients in clinical trials to other drugs. And then another lesson for the future, the EU research programs were disconnected from clinical research funding of the member states, and there were no incentives by the member states to participate in the EU-funded clinical trials. Sixth, about EU funding. I think the EU needs to be applauded for the flexible and the very rapid response to provide funding. But we could not jumpstart because there were no pre-established pools available. And so, again, it was one of the first organizations to release funding for clinical research. Nevertheless, for instance, during the month of February, we had to spend a lot of time on grant writing. And then the top of funding of Recover came in June 2020 with another 50 million euros. And that, of course, created a great boost to REMAP-CAP. And it was just in time for the second wave. Would it not have been wonderful if we had received this top-up funding already in February, and if you could have focused already in the beginning of February on clinical research, on protocols, on patients enrollment, etc. So there remains a need for an immediately available epidemic response fund, which could be rapidly deployed without the need for open calls for proposals and peer evaluation process. 7, academic cannibalism. This was a term used by one of our colleagues when we were discussing the lessons learned. And there was indeed academic cannibalism because there was a huge competition between hospitals, between researchers and clinicians to enroll patients in the clinical trials. And I saw it also in my hospital. People were really, let's say, competing with each other for clinical trials, for enrolling patients, and this was really not good. Most trials probably did not even reach their target number of inclusions and therefore failed to deliver solid conclusions. And thousands of patients were treated for COVID-19 with drugs that were not registered for its indication. The so-called compassionate use is a term often referred to by industry, or we would also refer this to compassionate misuse because there was no collection of high-quality data. 7th, interestingly, most of the pharma-sponsored clinical trials were relatively unsuccessful. And none of the therapeutics currently recommended in Europe hospitalized patients, such as steroids, remdesivir, or tocilizumab, actually have been recommended as a result of data from pharma-sponsored trials. And this may be because the model adopted by companies was a series of modestly sized trials built among single agents as opposed to the more ambitious adaptive platform trials. So that's a lesson in history that, in fact, the recommendations for some of their drugs will not come from their own sponsored trials, but it will come from publicly funded trials. Then about clinical trial design. I think it was clear that there was a great value of having perpetual observational intervention studies. For instance, MERMAID ARI enrolled patients right from the start. And as I said, REMAP-CAP proved its value for efficient and rapid epidemic clinical research response, which enabled indeed simultaneous and sequential evaluation of multiple treatment regimens. And in fact, it became a very effective trial where fewer patients allowed us to reach interpreted results. So we should really, in the future, continue these observational platform trials, particularly also in primary care. And this is where I think we failed in 2020, because unfortunately we do not have, and we're not able to build a platform with responsive data randomization in primary care. Unfortunately, I should say, because Chris Bertley in the UK was coordinating a trial, principal trial, that we could have rolled out over the rest of Europe. And with 10-15 million euros last year during the summer, we would have by now had an adaptive platform trial in primary care, and we would have been able to do some great research, answering some very important research questions about optimal management of patients in primary care. So that, I think, really was unfortunate. We do not yet have such a platform trial in primary care. And then last lessons about our own vision. I think we had a lot of discussion over the last 5 years developing such a sustainable, operational, high-quality, large-scale clinical research European infrastructure. And we were doing this, and we wanted to do this by combining experts on antimicrobial resistance, mainly bacteriologists, and experts on emerging infectious diseases, mainly virologists. And a lot of people were not following exactly what we were saying, and couldn't really see how you could merge these two different, let's say, disciplines. But I think COVID-19 proved that we were right in our vision to build such a network, and proved that the EARL issue, the ethical, administrative, regulatory, and logistical challenges and problems to be addressed by both experts and research infrastructure have indeed more overlaps than differences. So I'd like to finish with my favorite lesson. And that is that, and it would have not been a big effort to do so, but if the chief medical officers and the ministers of health, like in the UK, if those ministers of health and chief medical officers in the EU had supported top-down the perpetual observation and intervention status of Prepare and Recover, and if there would have been immediate substantial funding provided to our Prepare and Recover trials already back in February 2020, then I think we would not have missed the bulk of the first wave. So that to me is really one of the most important lessons. You need this top-down support in the European Union to become successful exactly the way the UK has done this. And so therefore, I think it's extremely important that member states and the European Commission should develop a partnership for several reasons: to leverage funding for clinical research during pandemics, to plan future research priorities, and then finally to disseminate this well thought out planning by the public health and clinical leadership. So in conclusion, it's been a great journey, if I may say, from let's say a clinical research perspective. We've learned a lot of lessons. And so this is now really a once-in-a-lifetime opportunity to gear up these public and private resources already invested in Compact and Prepare, as well as in many other funded EU COVID-19 EU projects, such as Recover, EU Response, or AUKUS of Accelerate, and several others. And really, if we don't collaborate now, we will fail and stand accused of having turned our backs on the prospects of tackling future infectious diseases, because this is not the last one. There will be more pandemics for the benefit of European citizens and also citizens across the globe. So many thanks for your attention.
Thanks very much, Herman. Am I correct that I'm now following that superb presentation? My name is Nina Goba. I'm a senior researcher at the University of Oxford, and it's my pleasure today to be joining this panel to be talking about rapid deployment of clinical research in public health emergencies. And I will be drawing on work that we've taken forward since the start of Prepare in 2014 that relates to the ethical, administrative, regulatory and logistic issues that stand in the way of rapid deployment of clinical trials, as well as other forms of clinical research in a public health emergency. And I'm really going to be focusing on what it is that we learned during the preparedness phase of the clinical research response through our years working in Prepare. But I'd like to acknowledge the team of researchers really who've been working on this work package, in particular that led by Alistair Nicholl, who presented earlier in the webinar series. It's really been a team effort in taking forward this work. There's been an active program of work since Prepare started in 2014 to identify some of the key structural bottlenecks and to propose, and where possible, to implement solutions. The rationale for this work that has been mentioned many times now through the Prepare webinar series and by Herman now this morning was the missed opportunity for research to inform response in any meaningful way during the H1N1 pandemic. And so when we started our work in 2014, we really began by looking to understand where the delays and the bottlenecks were in relation to rapid deployment of research and what experts saw as important solutions to these challenges. But of course, the work that we were doing at the time was unfolding against a broader context of emerging infectious health threats in many parts of the world, where the integration of research, including clinical research, was increasingly becoming a feature of response. And together with this came a number of high-level global advocacy initiatives and mechanisms to accelerate the process of research and development for infectious health threats. And it's now, as we all see, well accepted that research and evidence is a core part of contemporary responses to public health emergencies. But there were some key moments and some key aspects that really sort of, I think, has accelerated some of our own thinking. For example, the experiences in response to the Ebola epidemic in West Africa, where clinical trials were really pioneered in tough epidemic settings, and some key lessons that were learned from this experience and captured in a report from the National Academy of Science, and also some high-level normative guidelines that were released from WHO, for example, around managing ethical issues, including for research, as well as the good participatory practice guidelines around involving communities in research design and delivery. And the Global Preparedness and Monitoring Board was launched in 2018, and in their inaugural report in 2019, they highlighted how the world was unprepared for a rapidly moving respiratory pandemic threat. And in that report, the GPMB also highlighted the need for increased and sustained investment in research infrastructure in anticipation of such an event, which of course is what faced us all at the start of 2020 with COVID-19. So this was sort of the context against which our PREPARE work was unfolding. Our work, of course, was focused very much on Europe, in particular on EU member states and associated countries, and primarily on readying our clinical research networks for a response. And we based a lot of our learning and conducted empirical work on PREPARE clinical studies to better understand things like timelines for approvals, contracting, and so on, as well as to understand the experiences and views of research staff and patients participating in these studies and their anticipated concerns about delivering similar research or taking part in similar research during a pandemic. So our focus was really around publicly funded phase 3 trials and observational studies. So Herman has presented the slides on the different outbreak response modes, and one of our focus was really on readying the research networks for rapid deployment for a phase 3 response, which is what came in the form of RECOVER. And in preparation for this, we identified a wide range of barriers and solutions, and they're listed here, and I'll talk to these now. But we also developed some operational tools that we anticipated would support our research teams in delivering a phase 3 response. But I think one of the lessons that we've learned with this is that while many of the tools were useful for preparedness, they were actually less useful operationally for informing response. So, for example, we mapped where in Europe there was provision for fast-track ethics and other regulatory approvals. We surveyed members of the research ethics committees and regulatory authorities about their perceptions about how ready they were to respond to a pandemic threat. And we identified a number of significant gaps in preparedness. But when it came to response, these maps quickly became redundant because of the highly dynamic and fast-paced nature of the emergency, but also the space for innovation, I think, that arises at the point of crisis that can't really be anticipated or mapped in advance. So many of the gaps that we had identified earlier were suddenly filled because there was an immediate sense of urgency and a need to do that. I think the other aspect that we estimated and of course had less direct influence over were the political barriers, for example, the disconnect between EU-funded studies and those that were nationally driven. But many of the higher level solutions that we identified over the years are now, of course, recognized as best practice for expediting research responses. Many of these have now been tried and tested for COVID-19, not only in our research consortium, but in other settings as well. And in particular, we highlighted the need for rapid release of funds at the point of outbreaks, prioritization of research questions. There are, of course, multiple complexities in implementing this kind of solution and many more detailed lessons to be learned, I think, through efforts to deliver this kind of solution. Pre-positioned, pre-approved sleeping protocols, of course, widely recognized as one way to speed up deployment of research. In our preparedness work, we identified the importance to fund maintenance planning for these protocols and to ensure that there are simulation and activation testings that can happen and that these protocols can be delivered against active clinical research networks. When it comes to novel trial designs, PREPARE has, of course, championed this design in REMAP-CAP and also in ALICE. And I think one of the lessons for preparedness is that the complexity of these trial designs really needs time to conceive, deliberate, and to develop the intellectual architecture of the study design. And all of this really has to happen in the preparedness phase. So, for example, a lot of the thinking that went into the ALICE trial during the preparedness phase, ALICE was a pragmatic, open, response adaptive platform trial, meant that that same design could then be used in the form of the principal trial, which is, of course, active in the United Kingdom. Some of the work we've done in PREPARE has been to focus on communicating the complexity and elegance of some of these designs to a lay audience. And I think many of the tools that we prepared during the preparedness phase were able to be rapidly updated and adapted for use during the response phase. The clinical networks, the active clinical networks, of course, are absolutely key to rapid deployment of research. And I think the social capital that gets built again over time is what's key here. It's in many instances, it's the relationships and the trust that gets built with national and local principal investigators and research staff, and the training and the capacity development initiatives that have been underway for some time that allows for effective leveraging of these networks. So again, sort of a strong focus on the investment that happens in preparedness phases, but really the output being the social capital that gets developed. In terms of ethical and regulatory approvals, I've already said, we did a lot of work in this area, and we actually found that in many instances there were innovations, bespoke mechanisms being put in place at national level, in particular for nationally prioritized studies. I think at local levels there was more variability, particularly to do with fast-track reviews and also likely to do with capacity issues, but also some interesting models of practice around how principal investigators were supported in thinking about the ethical design of studies. For example, one of our investigators had a dedicated ethics review board advising on the study and had the ability to have informal conversation with the chair of that group. When it comes to contracting, this was really one of our biggest hurdles. If we look at the time taken to contracting and clinical trial agreements with different sites, we proposed a number of solutions related to pre-agreements or generic contracting, but we made very little progress during the preparedness phase. I think simply there wasn't the urgency to find a solution to this bottleneck. During COVID-19, there was a solution that was found to have some progress to go forward based on a letter of intent, but nevertheless, this was a key bottleneck and remains a key bottleneck to rapid deployment of research. In terms of innovative approaches to participant identification and recruitment, we did a lot of work and thinking about this in the preparedness phase, particularly looking at public view adaptations to research enrollment procedures during a pandemic. We ran consultations, we ran focus groups, we conducted a public survey to better understand public views, and found in general strong public support for conducting clinical research in pandemics and appetite for adjusted research enrollment procedures to allow these studies to go ahead. But I think, again, this is an area where it was quite difficult to preempt what the needs were and what the local and contextually relevant solutions might be. And I think we've seen a lot of innovations in this area too. So, for example, the Principal trial, which is based in primary care settings in the UK, ran into a challenge quite early in the pandemic when patients were told not to present to doctors and consultations started happening remotely. And of course, that had an impact on plans for patient recruitment. And the team also wanted to make access to the trial available to anybody who's eligible rather than only those who could receive care at participating sites. So the trial team set up a process of central recruitment, including self-referral with online consent and telephone follow-up. And the outcome of that was recruitment of a more diverse and nationally representative sample of 4,756 patients who were randomized. And I think these are the kinds of examples that we really want to document and learn from going forward. And some of our preparedness work also highlighted the need to pre-position communications materials and to pre-position mechanisms for rapid public involvement and engagement. And I think the communications gap was really highlighted in one of our simulation exercises. We recruited a strong team in Prepare who was then able to develop a communication strategy in Recover with a focus on becoming a trusted source of information to counter in a small way the flows of misinformation and disinformation, what is now termed the infodemic for COVID-19. Engagement or involvement, and I think globally, stronger advocacy for patient resources very early through the research design, even in a public emergency. We did have a bespoke public panel to advise Prepare researchers, and our communications team also assembled an EU-wide public contributor panel via social media to feed into the development material. But I think more really can and needs to be done in this area. We do see some great examples in other parts of the world, particularly from those from civil society organizations that have been involved in HIV trials. And in the UK, where there's a strong policy context of public involvement in research, a bespoke solution was set up to match research teams with a centralized public panel. So again, some good lessons there that we can learn going forward. But in retrospect, I think another one of the unexpected outcomes of the engagement work that we took forward in the preparedness phase is that it started to socialize the idea of social and behavioral sciences in a very biomedical arena and a very biomedical research consortium. And I think there's potential further with the contribution of social science research in its many forms in strengthening clinical research responses. This is also something that's been raised in the Global Preparedness and Monitoring Board report. But I think one of the legacies of PREPARE, as well as from its predecessor and sister networks like GRACE and ValueDx, is that in Recover we now have a team of social scientists who've been working to deliver a cross-cutting program of work, leveraging the clinical research networks to understand experiences of patients and health providers in clinical settings, as well as in home-based care. And I think the social science research could be rapidly deployed because of the multidisciplinary nature of the consortium, but also because of the relationships that have been built over time and because of the leadership from Herman and Sylvie van der Werf to make space for that kind of research to be conducted. So looking ahead, and I don't have a final slide on this, but my key takeaways are, I think that we have learned more about what is needed in the preparedness phase to launch a rapid research response in a public health emergency. I think we've learned that there will always be unexpected opportunities, and a need for flexibility and agility is key. A cornerstone for rapid research response at scale, I think, is the trust and relationships, the social capital and the intellectual capital that gets built over time. I think we need metrics for identifying key dimensions of research preparedness at many levels. The frameworks that allow for assessment of national pandemic response don't include strong metrics for research preparedness. And I think we need more and sustained focus on communications and engagement with citizens and with civil society, not only to support efficient and effective delivery of trials and clinical research, but also to inspire future generations around the power and potential of science and research and development. And with that, I thank you for your time and attention, and I hand you back to Herman.
Thank you very much, Nina, for a fantastic talk. Very impressed. It shows the beauty of PREPARE and the multidisciplinary nature of the PREPARE project, and it's been very rewarding and stimulating and exciting to work with you, Tina, as a social scientist, and many other members of the social sciences team. So thank you very much for all the hard work that you've done over the last 7 years of PREPARE, and once again, it's been a great joy to collaborate with you, and I hope that we will continue to collaborate in the future. Thank you, Nina. All right, so then it's also my really great pleasure to introduce the next speaker, who doesn't need any introduction, of course, Marion Koopman. Marion is working at university at the Erasmus University in Rotterdam, and she's of course an extremely well-known person in the field, and it's been also fantastic to collaborate with Marion, to have her on board to PREPARE, and so looking forward to her talk, and she will be talking about the diagnostic preparedness and where do we stand now. Marion?
Yes, thank you, Herman, and thank you for your introduction. Can you hear me all right? Yes?
Yeah, we can, yeah.
Okay, yes. So, yes, I will talk briefly on where we stand with regards to diagnostic preparedness. And this is a challenging question that we have discussed from the start of PREPARE, in how do you prepare clinical researchers, but also public health arena for emerging diseases when it comes to diagnostics. How do you prepare for a disease that you don't even know yet, when you think that an occasion like this is really too rare for individual practitioners to come across? Having broad preparedness is too costly, and there's so many potential threats. So where does that start and where does that end? If you look at the diagnostic landscape, we look at it as an iceberg, where there's the tip of the iceberg over the water, which is the routine diagnostics in place that differ depending on the level of the setting, typically focusing on the well-known and treatable conditions that we want to know about. And this is a diagnostics where there's a lot of routine available, including commercial applications. Then you have the bit that sits under the water, which is reflecting the broader differential, including more unusual presentations, but still of the known. where these are less broadly available, maybe in reference settings, often in-house assays, and there's a limited market. And then there's the deep unknown, which can have these emerging infections, exotic infections, with all sorts of methods that are poorly standardized and even may pick up infections for which the clinical relevance is yet to be determined. Now, the importance of diagnostic preparedness, I think, is very well illustrated with the experience of Zika virus. As you recall, this was a declared public health emergency of international concern in 2016, after Brazil had sounded the alarm, noticing that there was an increased wave of births of children with severe birth defects, microcephaly particularly, but also other neurological syndromes. This was caused by a virus that until then had not been detected in the Americas, but from later genomic studies, it was constructed that this virus had already been introduced 2 years before, so had been circulating under the radar until it really exploded into this massive outbreak. So that begs the question, can this also be happening in Europe? In Europe, we're looking at a range of arboviruses that we know, particularly tick-borne encephalitis, West Nile here in the south, and Alcorma in a very small pocket, and then viruses that are zoonotic but do not seem to cause very significant disease but are present in wildlife across Europe. And there are, so if in terms of emerging disease problems, there are projections that arbovirus problems will increase in the European context as well. Now, and some similar observations were made here in that we have hidden arbovirus circulation in Europe, similar to the Zika virus example, where Usutu virus was found widespread in birds, so not people, in birds, already circulating endemically since 2010. Whereas over here, 8 years after that, the first Usutu virus positive blood donors were found. Now, this is not a big disease problem, but Usutu virus can infect people, can cause neurological symptoms, and the message is also in Europe we have diseases that may be going under the radar. So this has also brought about the thinking about how can you be better prepared and link up with the animal side of surveillance and diagnostics, because that's where many of the new diseases come from. And this is a preparedness summary that we wrote as a Prepare Consortium to illustrate that you may have considerable virus circulation in animal reservoirs before picking that up in people, and that also calls for collaboration between groups focusing on animal diseases, like and those are in other projects, and that's what's been built into the Next Clinical Research Network. Now, there's also clear successes. If we look at, Herman already mentioned this, the response to the COVID pandemic, where the first patient here was the first cluster was notified by colleagues in China, who then also characterized the etiological agent in very, very fast turnaround time, and importantly shared the first sequence of the virus around January 10th. And that's when the group led by Christian Drosten with several partners within Prepare managed to, within a very short time frame, validate the first diagnostic PCR assay that was also published on the WHO website, and it was used to prepare kits for diagnostics that WHO shipped across low-income countries, diagnostic preparedness. So that really was a success. And a big contribution to that success was that there was already a comprehensive repository of clinical reference specimens from the studies that had been done through PREPARE and its predecessor projects. Very important and much ignored fact. And this having this established collaboration in place really allows for very fast forwarding. Now, the next phase was considerably more and is considerably more challenging. So the initial assay design we've seen is really not the biggest hurdle, and that the capacity for that is there. But how do you then, from those initial assays, go to widely deployable techniques, given the lack of standardization across labs in a clinical network, but also beyond that? Here's just an example of a couple of platforms that have been used with an explosion of commercial assays being developed, with difficulties in continuing to build biobanks in the competition that Herman just mentioned. And all of the individual assays then really need some kind of fit-for-purpose validation, and there was really the lack of a coordinated process here that I think has caused considerable delays. Now, what's also been a key lesson is just how important genomic information is. That have been looked at, but more in research projects. So this is a slide that I've used before, just explaining how important genomics can be and what you can learn from adding on genetic information to the diagnostics. But this is now an image that was used in Parliament, in the Netherlands in this case, just to explain to politicians why this is so important, what you can learn from genomic sequencing in an outbreak investigation. This, in this case, looking at the spillover or spillback of SARS-CoV-2 in MIC. And for any future activity, it's clear that genomics need to be embedded in any work further. Now, that had already been ongoing also in the different mermaids trials that were going on in the PREPARE network. For instance, here looking at Zika virus and other arbovirus genomes, including sequencing support in different countries at request. So in China, in Brazil, in Suriname, here from teams doing that work. And similarly, what we have been seeing and what I'm sure will continue to be further developed is the importance of serology in this, not so much as patient diagnostics, but because of all the other additional information that you can learn from antibody testing into your patient evaluation. It talks to you about prior exposures, possible protection, breakthrough infections that we're looking at right now for SARS-CoV-2, for instance. And we've learned through combinations of serological testing and virological testing what are markers of infectivity. And here's an example of the type of multiplex serology assays that are developing. This was developed through PREPARE for a full battery of viral respiratory pathogens for the mermaids studies. And an example of an application here in the COVID pandemic, where you can then see that patients with severe infection, the red bars here, have clearly higher levels of antibody responses to SARS-CoV-2 antigens, not to the influenza, which is added as a control antigen, but that there's also some boosting of antibodies to other coronavirus, seasonal coronaviruses. And there's a line of studies now trying to understand whether or not this type of boosting may interfere or may have some impact on the course of disease. So key messages that I want to leave here is that rapid response to an emerging infectious disease does require a prepared diagnostic network. There is an invariable gap. We will not be able to solve that of several months before validated assays for common platforms are available. And that is something to think through how to prepare for that. There is no clear-cut delineation between clinical and public health diagnostics, as many key public health questions can be addressed only when looking in patients. And syndromic diagnostics, including genomics, are here to stay, in my view. And of course, the well-characterized biobanks are crucial for all future efforts. And this is to acknowledge the partners within PREPARE that have been working mostly on the diagnostic aspects of the support for the trials.
Thank you very much, Marion, for a really, really great talk on where we stand today with diagnostics. Really, really great. And also thank you very much for the wonderful collaboration. It's really a wonderful journey in PREPARE, and I hope we will be able to continue for many more years, despite our age, Marion, that we can continue to collaborate successfully.
Age is just something in your mind, Herman.
No. Good. Thanks again, Marion. We'll come back to you during the Q&A. Okay, so then we move on to the next talk.
It is my pleasure to give the final presentation in this webinar series from PREPARE, and I will talk about the future of clinical research in Europe, ECRADE. And ECRADE stands for the European Clinical Research Alliance on Infectious Diseases. In 2017, Oosterholm and Olshaker stated that there are only two infectious disease situations that can be considered inevitable serious pandemic threats: influenza, or let's say a viral infection, and antimicrobial resistance. Of course, PREPARE was addressed to dealing with future pandemic threats, mostly viral infections, but the threat of antimicrobial resistance had been recognized before as well. About 10 years ago, the Innovative Medicines Initiative designed a program called New Drugs for Bad Bugs to deliver a pipeline of new antibacterial agents for patients in Europe. They created a pipeline from early discovery to phase 4 and projects that addressed specific aspects of this pipeline of delivery of new antibiotics. And in green, there are 3 projects named COMBACT, COMBACT-NET, COMBACT-CARE, and COMBACT-MAGNET that address the clinical evaluation of new antibiotics. The objectives of these COMBACT projects were to create a self-sustaining antibacterial development network in Europe by expanding clinical research and laboratory networks, to optimally align clinical trials with investigator sites, and to obtain clinical and epidemiological data. The goal was to increase efficiency of antimicrobial drug development by aligning these clinical trials with cutting-edge molecular methodologies and trial design, and to deliver clinical trials with various candidate compounds from the FPR partners that participated in these projects, such as MedImmune AstraZeneca, Arcturus, AstraZeneca, Davoltera, and the Medicines Company. So COMBACT started in 2013 and is still ongoing. In 2014, we all know that PREPARE started, and that ended a few months ago, and some of these activities are continued in RECOVER. And then we have ValueDx that started in 2019. So while these projects are still running in parallel, we foresee for the future that these activities will be merged into what we call ECRADE, Again, it stands for the European Clinical Research Alliance for Infectious Diseases. The purpose of eCRATE is to reduce the impact of infectious diseases on individual and population health. And the vision is to efficiently generate rigorous evidence for new or improved diagnosis, prevention, and treatment of infections, and to better respond to infectious disease threats. This is facilitated by a European multidisciplinary clinical research network and innovative research approaches. So the strategic agenda of eCRATE is depicted here. From the left upper corner, we can start. It aims to be a sustainable clinical research network that is built on past successes, that will be financially self-sustainable, that builds public and private alliances, and creates global synergies. And if we then go from left to right, on the bottom of the slides, it's built on a community of networks. It encompasses all clinical trial aspects. It aims to generate evidence to improve patient care and aims to be innovative and flexible. Depicted in another way are the different aspects of eCRATE that we see. In blue are the clinical sites, the networks that we have, such as networks focused on pediatric infections, clinical trial networks in hospitals, but also in primary care and also in long-term care facilities. Then we have the laboratory facilities in LabNet and in BioNet with biobanking facilities. And then the scientific support coming from EpiNet on epidemiological research and StatNet focusing on statistical research. We envision that this all will be enabled by a WORM-based clinical research network. And this European WORM-based clinical research network comes from the networks that were established in the projects that I mentioned. So the 300 primary care sites in 18 European networks in 18 European countries, about 1,000 hospital sites in 42 European countries, 90 pediatric sites in 18 European countries, and 850 laboratories in 41 European countries. All these networks are joined in eCRATE, and we aim there to have an organized, prepared, engaged, resourced, active, trained, extensive, and sustainable organization. I think most of these aspects speak for themselves. Organized is that we have networks with an organizational structure combining all the networks. The networks will be prepared to deliver the clinical trials. They will be trained. They will be sufficiently resourced. They will be engaged by perpetual observational studies. Our services will be extensive, and the overall aim is to be self-sustainable for the future. So most of these networks come from the Comeback network. As I mentioned, these are about 1,000 hospitals in all the European countries. And these 1,000 hospitals can be roughly distributed in 3 types of hospitals or clinical sites. Those that already participated in clinical studies in the last 10 years, they were selected by site selection processes and already proved their capability of doing these clinical studies. Then we have member sites. They participated in site selection processes but have not yet been selected for any of the studies. And we have prospective member sites that have shown an interest to become member of the network. At this time, this is the number, these are the trials that are currently ongoing in the network. Some are part of the Comeback project, some of Recover, also specific COVID-19 trials and trials outside that spectrum in green. In total, more than 35,000 patients by now have been enrolled in all these trials in this network. Now, on the next slide, we see the trials that have already been finished in the last years. So these were the completed trials. We aim to drive innovative clinical research through a broad portfolio of projects. And here we want to use innovative design elements in the clinical studies. For example, create studies with continuous enrollment. to allow shared control groups in clinical studies, where possible to use response adaptive randomization, use master protocols, and build nested trials in perpetual observational studies. And finally, we also want to establish new platform designs, as we have seen for REMAP-CAP. This is how it looks. We have these perpetual observational studies that serve as a basis for other studies that can be nested within such an observational study. For example, in ICU, we will establish a study for patients at risk for ventilator-associated pneumonia, an ongoing study with continuous enrollment of eligible patients, and in such a study, other studies can be nested, such as randomized controlled trials. The same we aim for complicated UTI, acute respiratory infections, and disease X, unknown infections, for example, with patients based in the ER. In primary care, we aim to establish such a network and such a perpetual observational study for acute respiratory tract infections. And new infections in new population can be added if we have the resources to do so. So for this year, we aim to establish these observational studies for VAP, QT, which is complicated urinary tract infections, acute respiratory infections, disease X, and acute respiratory infections in primary care. In addition to that, we already have the REMAP-CAP study and the EU RESPONSE study, which is in development, also an adaptive platform trial for evaluating treatments of COVID, has expressed an interest to do this under the umbrella of eCRATE. For these observational studies, we have specific targets for the coming years. For the VAP study, for instance, we want to have 40 ICUs in at least 15 European countries that enroll about 4,000 patients per year in the observational study. And that cohort can then be also used for nested randomized controlled trials, for example, or for building an adaptive platform trial. The numbers of these other observational studies are listed here as well. Next slide. This work is already ongoing, so we have studies here in red that are already set up and that will be continued towards the perpetual observational studies, and that will be nested within these perpetual observational studies. So with eCRATE, we want to add clear value to stakeholders across the ecosystem. Here we can see where eCRATE is positioned in relationship to industry, regulators, public health authorities, academics. We have clinical research sites and other research projects, and this is how we want to serve the European health care system. And the ultimate goal is, of course, to make clinical research more efficient. We have seen the dramatic effects of increasing efficiency in developing and evaluation of COVID vaccines. The same we want to do for other treatments. And here we see where we think that we can create benefit and can increase the efficiency of clinical evaluation of new antibacterial agents, but also of clinical evaluation of new diagnostics or new prevention strategies. We think that we can reduce the time for clinical evaluation by at least 25%. So how do we establish eCRATE? Well, the 3 steps towards eCRATE is first eCRATE Base. eCRATE Base is an Horizon 2020 project that has been awarded and just started 2 months ago. And this continues some of the activities in Prepare, Comeback, ValueDx, Recover, and eCRATE Plan. As part of eCRATE Base, eCRATE will become an independent, non-for-profit legal entity this year. So eCRATE Base coordinator is UMC Utrecht, but the plan is to create an independent, non-for-profit legal entity, and that one that will then serve as the coordinator of eCRATE Base. And eCRATE Base is then the initial set of activities for finally eCRATE. eCRATE Base, of course, is organized as a classical large European project with work packages related to the organization and management, but at the heart of this project are the clinical studies, work packages 2 till 7, which are the perpetual observational studies, and REMAP-CAP, the laboratory facilities, and the data management support, including EpiNet and StatNet. So the eCRATE headquarters is currently in Utrecht, and it probably will be there for the coming years. But also we have eCRATE chief offices located across Europe, as you can see in this slide. And on the next slides, we see the pictures of the persons belonging to these head offices depicted here. Many of you will recognize from their role in PREPARE, such as Marion Koopmans, Lenny Derde, Peter Horby, Chris Butler, let me see who was Carlo Giaquinto, Jacques Demotte, and all the others, and of course Nina Gobat. The others had a very important role in Comeback. So there's really been a merger of the PREPARE and Comeback activities, and Herman Goossens and myself, together with Frank Degen, will act as the management team of eCRATE. With that, I would like to thank you for your attention and ready to take questions after this presentation.
Thank you very much, Mark, for this great and very inspiring talk. It's been a wonderful journey also to work on this eCRATE concept, and as I also said before, I think we've proven that it was the right way forward, and we also hope, of course, that the Commission, European Union, the Member States will go along these lines and fund such an infrastructure, that we no longer depend on ad hoc funding, but really that we can build such a sustainable infrastructure with public, but also, of course, with private funding. And great to collaborate on this with you, Mark, and also many other members of the family. And of course, we hope that the family will expand very soon, and that more people and more colleagues will join. Now, there are a lot of questions, and we only have 2 minutes left. So unfortunately, I don't think we'll have time to address the questions for Nina, for Marion, and for Mark. So what I'm going to do perhaps is I'm going to just select 1 or 2 questions from Mark, and then I think I'm going to have to close this webinar. And I'm going to close the webinar because Jeremy just sent a very, very long message. Unfortunately, he had to leave, but he sent a very long message. I haven't read it yet, but I will happy to read it to you, because I think, as usual, it will be a very inspiring message for all of you. So let me first address the question to you, Mark, and then I will finish this webinar with Jeremy's message to us, but also to many colleagues who are online here today. So, Mark, the question is, what are the elements that probably contributed to the delay in Europe relates to the silos of health versus research? And research preparedness really should be part of overall pandemic preparedness activities. This link between health and research is functional. So what do you think about that, Mark?
Yeah, that has been very, very obvious that was a problem, especially in the first wave, where the research activities, especially the clinical research activity, were really hampered by the overwhelming health requirement that physicians were put under. So many of us wanted to do studies and wanted to start up. And then the other thing was that it was unorganized. It was fragmented, and everyone was running through each other, running across each other, but not really collaborating. And I think we have seen a few examples where there was clear success, one of them being REMAP-CAP, the other obviously being recovery. And then the mechanisms for the success are very clear. There needs to be coordination, there needs to be preparedness, and there needs to be also kind of a structure, maybe a top-down prioritization of saying, okay, this is what we're going to do first. First, we're going to do this intervention, and then we can think of others. And we need to collaborate in order to have results fast. It's not of any use to do a trial that gives us an answer in 4 years. We need it in 3 months. So that really should make people think differently about clinical trials. This is not the way we used to think about clinical trials. The rules now are different. So we really must learn from that. And I think we did learn from it. But it's very, let's say, embarrassing to say that we still had to learn it again.
Yeah. So maybe just one short last question, and then I will close the webinar with Jeremy's message. Again, for you, Mark, I mean, you're a great researcher, Mark, and this refers to the academic cannibalism, and you and I know where this comes from, and you use this for the first time. So this is a question about, indeed, research, doing this research and competing. And so the question is, should we change or restructure research incentives or research rewards, Mark?
Yes, that should definitely, it should not, the research, especially if you do multidisciplinary international research with many people, it cannot be that all that collective work is the pure gain of one or two persons. It's the collective group that should be seen as the ones who did that. And I think that's also, let's say, recognized increasingly by the scientific field. And therefore, whether you have, people think if I do my own 200 patients, may get me a big publication rather than have my 200 patients in a 2,000 patient study where I will be somewhere in between, maybe. And that needs to change, that thinking, especially in a pandemic like this, because we're all in this business to help patients. And the patients are best, let's say, served with rapid and high quality answers. So that's definitely something that needs to change, and probably that change will come slowly, but I think the pandemic is a perfect example of why it should change.
Okay, thanks, Mark. Yeah, I'm really sorry, but we have to close this webinar. There are many more questions coming in, and I'm very sorry, but we need to close. And I'm going to look now at the message that Jeremy just sent. Again, I'll read this to you. And so this is what he says. Great to be able to join, but I will have to drop off in a few minutes. Just thoughts first. Thank you for the invitation and kind opening words. Herman, good ideas are easy. It's implementing good ideas that is so difficult. Second, still a bit frustrating to conclude. We have learned a lot of lessons and we will be prepared better next time. We have been thinking about this as a world since, certainly since SARS-1, 17 years ago, exclamation mark. Third, we must not fall into the mistake of saying we did okay. REMAP-CAP, recovery, and some others deserve great credit, but a lot of clinical research around the world, including in UK and the EU, was not done well. Fourth, must never underestimate politics, exclamation mark. Politics are at the level of politicians, but also politics of clinical research and scientists. Fifth, missing the first wave when EU-UK had more than 1 month to get ready has to be unacceptable. Sixth, EU-UK has to have the train tracks available, working all the time. Protocols on shelves is not enough. Needs everything in place and active all the time. It cannot depend on people writing grants. 2 exclamation marks. Has to be an immediate flow through from funders. By immediate, I mean within 24 hours. You would not ask the military to write a grant, would you? Sixth, really important, we all don't think how well we did. Need to retain the frustration of a system which did not work fast enough when it had to. Only with that honest assessment and frustration will reforms come, exclamation mark. Sorry to have to step away and look forward to joining the next photo and next meeting of the daughter or son of Prepare. So I think a lot of very important messages, and I think as usual, Jeremy was spot on for a lot of things. So I think this closes with his final words from Jeremy, and I think he deserves to close this webinar because, as I said at the beginning, he was really the father of Prepare Consortium, And so therefore, I think it's great that we could close this webinar with his final words. So thank you all very much for joining. It feels a bit strange because it's indeed our last Prepare meeting after more than 7 years. It would have been great to have this last meeting with a glass of wine on the terrace in the sun, but unfortunately that's not the case. But let's not complain and hope that we will have another opportunity to raise the glass to a really successful collaboration based on I should say friendship. And again, what makes a network successful is not the funding, but it's the friendship between people. And I think that's something we've all been able to enjoy in the last 7 years, and I'm sure that we will be able to continue to do so. So once again, from let's say my whole heart, thank you very much for the wonderful collaboration and look forward to the future. Thanks for joining.